Thanks, Nanopore, for having me today. Super thrilled to be here and share my work. Let's dive right in. So HIV is a retrovirus that infects CD4 T cells, and it integrates into the host genome. Once this happens, HIV behaves very similarly to a human gene. For instance, it can become transcriptionally active, which results in productive replication of the virus. In this context, HIV regulates its gene expression via splicing, which generates prodigious isoform ensembles and diversity within an infected host cell. On the other hand, HIV can also become transcriptionally latent. And this is characterized by lower viral transcription, no detectable viral replication, and the host itself might transition into a state of quiescence or a resting state. And beyond this general characteristic, very little is known about the HIV transcriptional program that leads to latency, which is a barrier to HIV cure, and it's a subject of our work. Now, to interrogate HIV transcripts that are present during latency requires really accurate amplification from very low amounts of viral RNA. To address this, we use a second generation of our MrHAMER protocol, which relies on dual UMIs that are incorporated during reverse transcription. These dual UMIs have synthetic priming sites that are retained during PCR amplification and subsequent library preparation. And once we sequence the samples, we can use our bioinformatic pipeline to cluster based on UMI sequence. And then we can collapse these into single molecule consensus sequences that not only get rid of PCR biases, but conveniently also give us increased single molecule accuracy, taking this from Q18 to Q35. We have validated this approach using both PCR-free datasets that we generated with the P2 Solo. And we've shown that our isoform enrichment is quantitative across at least 4 logs. And we have also used synthetic RNA mixtures that we generated. And we've also shown that we get expected values when using a range of different RNA input amounts. Now that we have validated the approach, we set out to ask the question, which is, what is the transcriptional signature of latency? And for this, we interrogated an HIV primary cell model of latency. And we did this both at the bulk latent population level, but importantly, we did it in T cell subsets. And some of these, specifically the effector memory and essential memory ones, are thought to be HIV reservoirs. And what we found is that there's significant isoform changes in envelope, VIF, and BPR genes. Importantly, because we also have high accuracy reads, we're able to couple these with isoform productivity analysis. And what we found is that some of the signatures that we've detected are suggestive of an immune evasion phenotype. So next steps, we're trying to dive in and really determine what the mechanisms of this might be and how it might affect splicing. And for this, we're coupling our MrHAMER methodology with RNA structure probing using DMS, which is a chemical that generates a mutational signal at sites that are conformationally flexible in RNA. In addition, we're coupling these with subcellular fractionation to understand whether there might be an interplay with nuclear export dynamics. So to conclude, hopefully I gave you how we're using a long-read RNA-seq as a window to interrogate RNA structure function. We've identified transcriptional signatures, and we're following those up with RNA structure function studies using DMS MapSeq, coupled with MrHAMER. And our goal was to integrate all of these findings into novel therapeutic modalities that either target RNA directly or that might be targeting RNA-protein interfaces. With that, I'll acknowledge contributors to this work and sources of funding. Thank you so much for your time.